Hello and good evening, everyone. Welcome back to My Aviofences and the European Fertility Society. This is our third event this week, so I'm really happy to be able to provide various topics. As you know, on Tuesday we were talking about PRP. Yesterday we talked about miscarriages and PGTA, and now it's time to talk about egg donation, epigenetics, and we have our special guest here, Dr. Omar Zamora is right here with us. Welcome. This is your first webinar with My Aviofences. I'm really happy to have you on board, and this topic is definitely fascinating, and I'm sure it's going to be an interesting session. How are you? Good to have you here.
Thank you very much for the invitation. I'm really happy to be here, and as you said, this subject is quite interesting. I had a lot of fun preparing this talk, and yeah, I believe and I hope that everybody will be as excited as I am after the talk. So thank you very much for having me.
Thank you so much for joining us. And yes, we will start, of course, with the presentation on our topic, but afterwards there will be time for your questions. Remember to put those in the chat section, and Dr. Zamora will simply answer them for you right after his presentation. Remember, it's all anonymous, so you can ask anything that's on your mind, and we are simply here to support you, to educate you, and help you as much as possible. And I just want to add that Dr. Omar Zamora, he's the fertility specialist at IREMA Clinic in Spain, and there are three locations in Spain when it comes to IREMA. So this is our first event. I'm really looking forward to it. I see someone from India is right here with us. Hello, and I'm sure there are other people coming from various places. So I'm really happy to have you here, and I'm really looking forward to it. So let's start with
Thank you very much. Very happy to be here. All right, very good. Well, thank you very much again to you, Caroline, to my IVF Answers and the European Fertility Society, and also thanks to IREMA, our team we work off, because they have also given me this opportunity to address this subject that, as I said, for me it's quite interesting and very fun. I have to say things up front. I want to apologize first because we have to reschedule this talk a couple of times, so I'm sorry for the inconvenience. Also, for my English, I'm not a native speaker, so any mispronunciation, please allow it. I'm sorry about that. And lastly, and most important, as I say, I'm a gynecologist. I'm a fertility doctor. I am not an embryologist. So if there's any embryologists out there, any biologists, maybe your eardrums will bleed when I talk about molecular biology. But I believe that, and I will try to make it very simple, very patient-friendly and gynecology-friendly. And as I said, the good thing about it is that it's a deal with patients every day. And this is a matter that it's a question that does come along very, very often in our consults. All right. So let's start talking about genetics first. All right. Before we talk about epigenetics, we need to have some concepts, some ideas clear about the basic functioning of genetics. I won't get too deep into this matter, but as I said, I want to address it. Remember, DNA, a very famous molecule, probably the most important one in our biology, and it's our source code. You know, we have 4 chemical bases there. They're paired, and according to that pair, we can translate it, an information. It's pretty much like the binary code that we may use in a software in a computer. So I repeat, most important molecule, and it has all the information. And the basic unit of that information, it's called genes, and it's like an interval, the shortest interval of DNA, and that it creates a specific protein, right? And those genes, it's like if we talk about a library and the whole genome, the whole amount of genes together is the whole library, and each gene, it's like a book that we take out, and it codifies a specific protein. And proteins are the way that living beings have to function, to deal with everything. So we are structured as receptors for hormones, as enzymes to catalyze and to make things change. So everything we do through proteins, and those are made out of specific genes for each one of them. And as I said, the whole amount of them is called a genome. And it's important to know that actually only 2% of the whole DNA, it's coding genes. When I say coding genes, it means that are sectors of DNA that produce a protein. So the rest of it, so pretty much most of it, it's just a structural thing. On it also works, and it's important for talk as a regulator for the expression of other genes. So I'm getting a little bit into subject with that. All right, important to know another word that is very common and unfortunately a lot of IV patients know of, and it's chromosomes. All right, and the gene, the DNA molecule, it's so long that it would be about 2 meters long if we could stretch it, extend it fully, and we have to fit that in a cell, in microscopic things. So it needs to be organized, it needs to be compressed, and to be organized in the genes or DNA does that through some proteins that are called histones. And they're like spool-like proteins where the strand gets, rolled around it and it gets very, very compressed. And the bigger picture of it, it's chromosomes, right? It's a larger structure with our whole DNA where it's organized as this. And we see it at some points during cell processes. And IVF patients know a lot about this because sometimes when they're not paired correctly, you all know that we have 23 pairs of chromosomes, and where they're not correctly paired is one of the main reasons of infertility, of miscarriages. That I believe that you recently have talked about this in my IVF, and it's something that, as I said, it's like the bigger structure where chromosomes are organized, right, through these proteins called histones. Just a little bit of information about how the proteins are made, are created. There are some enzymes that read this source code, right? In the library, they are looking for the book and they get the book. For this example, I will say, let us say the page, right? So they find the right page, and each page, let's say it's a gene. So they read that page and they make a copy. Like if you in the library make a photocopy of that page, and that copy is called RNA. And it's just one strand of the double helix of the DNA. It's just one of them. It's called RNA. In particular, this one is called messenger RNA, mRNA. It comes out of the nucleus to another place in the cell outside of the nucleus in the cytoplasm called ribosomes. And those are like the factories. And in that place is where the proteins get created. So that our messenger RNA sends the code, and there's a translation process where the ribosome starts assembling, it starts creating the protein for its eventual function. And one last thing about genetics, there are aspects to consider. DNA changes, and it has to change, and it's obvious that changes because if it wasn't like that, it would be just one cell at the beginning of the history of life on Earth. So it's changing, and it creates all the species and all the life forms on Earth. And it's, as I said, the foundation of evolution, those mutations, those changes in this source code. But it's important to know that those changes are random, right? And it's like a very basic thing in evolution and in Darwin theory, and that this happens randomly. And usually the body has the mechanism to try to repair those mutations, but yes, sometimes they still remain there, and sometimes they can be inherited, right? And they can change. But usually, and that is something also very important that we know, something very basic in biology, is that the somatic cells mutations, I mean, the mutations that you may have in any tissue, organ, or cell in your body, that we're not talking about all sides of sperm, so the liver, the skin, the brain, it shouldn't at all change anything in your germ cells. So those mutations that you unfortunately may have, because sometimes usually mutations means that you have high risk for example, cancer, something like that. And those things, fortunately, are not inherited in the germ cells. So germ cells have a way of protecting themselves and trying to maintain the best way they can their source code. All right? So knowing these basic things, now we're going to change a little bit and we'll talk about epigenetics, right? So as the word epi, you will think it's something above the genetics. So for me, the easiest way to explain this, it's I read this book recently about Garcia Marquez. It's an author that I really enjoy, Postmortem published, and they put some pictures of the manuscript, original manuscript, and some writing, some words that are crossed out, some and things that the author wanted to change, right? So that is a bit epigenetics. The source code doesn't change, but you cross out some things, or you fold the page so you cannot read that page. All right. So that is for me the most easy way to think about it. There's also an important doctor in this area. It's called Dr. Rechavi in Israel, and he says that this is a bit like the IKEA books. I'm not trying to advertise anything, but it's like this IKEA book that will tell you which furniture to be created. It has a mark, and in that mark you say, we're not doing this furniture anymore, or even the opposite. We might be not doing it for a while because it has that mark, and then we take it off and we say, let's start building this furniture again. So how does it work, right? There are pretty much three mechanisms how epigenetics works. The first one is, probably the most known one, as long as the second one that I will address, is DNA methylation, right? So there's an enzymes that control the addition to a methyl group. It's a very small molecule out of carbon and hydrogen, and it goes to a very specific part in DNA, right? In those chem bases. And in that area, they usually get added are areas that are clustered and they are grouped in, we call it islands in the start of the gene, in the promoter region of the gene. For genes or sequence of genes, there's always like a starter, like an engine where you start. So that moment when it gets activated, it unchanges the whole process. So it's very accumulated, those chemical bases in that place, the cytosine and guanine, and that is the place in cytosine where those methyl groups get attached. And when they usually do that, it means stop, do not create, do not transcript this gene, all right? The second one is histone modification. So those proteins that I told you that condense, compress the DNA, the spool-like ones, they can also change through substances that might give them, for example, methylation also, so adding methyl groups or acetylation of those proteins, and it changes the structure of it. So it might make usually the protein a little bit more dense, so it compresses even more the DNA. In other cases, more rarely, but it can also make it loose. But usually it compresses, so it doesn't allow, or it makes it more difficult for those enzymes that needed to read the source code to get access to it, right? So it also is a way to stopping making the gene and the protein. And the last one, and the most probably revolutionary one, most recent one, there's a Nobel Prize in 2006 won because of the discovery of this substance, are RNAs transcript, small RNAs, also called non-coding RNAs. They have several names, and it's just a very small strand of the RNA, and they work in a post-transcriptional process. That means that they are sent to the cytoplasm to block, to stop the messenger RNA that looks like this one. So it's like a final minute thing. So don't go there, don't go to a ribosome, stop making proteins, right? So, and it's very, very particular because it's believed, and there's a lot of evidence about this, it's not believed, it's like this, that those RNAs can be expelled from a cell. They can go into little vesicles, exosomes, and the cell can put them out of the cell itself to another areas of the body or of the surrounding area. So this is very important, what I just said. So what's the importance of epigenetics? This is a physiological process, right? So we have been understanding epigenetics for quite a while, and we know it's a process that helps especially make cell differentiation and tissue-specific gene expression, because every cell in the body has the whole genome. Every cell has the whole library, but they don't need the whole library. They just need a set of books. So through epigenetics process, the skin cells only use those sets, and the brain cells use another set for the neurons, for what it needs to do. So that is mediated usually through epigenetics, something also called genomic imprinting. This is, we always don't want to get into this too much, but we always get two genes out of mom and out of dad, the same gene. And sometimes we express one and another, and in some cases, we inactivate even a whole chromosome through epigenetics process because it's necessary for physiological and for normal development. As a matter of fact, there are also studies, I also say this because I think it's quite interesting, in which we find that cells in some tissues, especially I'm talking about the brain neurons, might express the whole amount of genes that it has only from mom and only from dad, and it can change during childhood. So that joke that you sometimes make when you're angry to that's your child, it's behaving, but well, yeah, it can be like that at some points of child. I keep on going. What we need to talk a little bit more is about the adaptive response to environmental factors in the individual that epigenetics also mediates. So maybe during your initial lifespan, in utero, especially inside of your womb, and in the first years and month of living, environmental factors, some external things can affect you. And then your body learns and it transcends during your whole lifespan because of that trauma, let us say, or that process that you may undergo. And that is mediated through epigenetics. And the most radical thing and the most revolutionary things, and it is a bit of a controversy because we still don't have that much of an evidence, and there are people that support this, there are people that are against it, is that it seems to be inherited. So you can actually, as a process of evolution, you can also make adaptation in your offspring because of the things that happen to you. And this is what we'll also talk about. Everything started, and like the most famous, the cornerstone of studies of epigenetics is one of the Dutch hunger winter study, right? There was an important famine in 1944, the end of World War II, and Nazis, as they do, did a very bad thing, and they blocked all the supplies of food to the northern part of Amsterdam, of Netherlands, I'm sorry. And it also was a very strong winter. Everything froze, so they weren't able to get food from anywhere. And they started taking less than 500 calories a day. A lot of people died, but a lot of people did it. And a lot of pregnant women were pregnant in that moment and had their kids during that period of time. And decades later, it was found that those kids that were born in that moment had a more probably a tendency to be obese and to have metabolic disorders like diabetes and problems with glucose and carbs and hypertension and even mental disorders. And I also have to say this because I don't want to forget it later. And it seems to also affect the next generation, all right? And this is not, the inheritance thing that I will later talk about. This is because of a Russian doll kind of situation. So you're pregnant, you have the baby inside of your womb, and that baby is developing germ cells during the pregnancy. So your future grandchildren are in there, and they can be affected by these external things. And this has also been seen in other cases. Not only Nazis did bad things, a lot of people did. So in China, during the Mao Zedong, during the China socialism, and there was a great Chinese famine, and also there was seen an increased diabetes rate in their offspring. Same in Nigeria and a blockage in Biafra. Also, obesity was increased. And even there are some authors that are talking about that, for example, the racial disparities that sometimes we may find in the risk for cardiovascular disease in African American in the United States, for example, that they haven't increased, it might be related to what they suffered previously. So, and not only from starvation or famine, it also seems to not be very good to over expose yourself. And there's a more recent study, 20th century, but with information from the 19th century, because Sweden has a good records. So they saw that at the end of the 19th century, in a very small town in northern Sweden, they were isolated, as a bit like the Dutch people, but they had crops and they were, doing their agriculture. And sometimes they had a good season, sometimes they had a very bad season, but they were isolated for quite a while. And that was recorded. And we have found out through records that the grandchildren of the families that were overexposed during 8 to 10 years old, those grandparents at that moment that were children, when they were exposed to a lot of food, to a feast moment after especially previous famines, they later had more risk to cancer and in general dying earlier. All right. And this all, what I'm saying has become like an hypothesis, right? And it's called the Barker hypothesis, or DOHA, developmental origins of health and disease. And that means that your tendency to be healthy or to have pathologies and disease starts very, very early, starts prenatal. And as I will say later, even preconceptionally. So it starts very early in life. And there are a lot of more recent studies that address this. I'm going to be as fast as I can with this. I don't want to get too deep into this, but it has been related to hypertension in offspring. I'm talking especially about nutrition, issues with food intake, right? It has also been related to food allergy, to asthma, to diabetes, obesity, as I said. Not only we're talking about things about food, but in epigenetics field, especially things about food, all right? But also other things like, for example, toxic habits like smoking. So we know that newborns out of female pregnant women that smoke during pregnancy increase their DNA methylation. We know that alcohol also during pregnancy has epigenetic modifiers in children and affects the circadian rhythm. That means the sleep-awake cycle that we all should have. Also, other drugs like opioids and parental abuse of a substance, parental substance abuse, I'm sorry, is related to these epigenetic changes. Sorry. In general, drugs, and I'm talking about medication also. So, sorry, some medication, especially for psychiatric use, has been related to epigenetic changes in children that could be correlated to autism, for example. So, quite important things. But not only it's chemical or let us say like everything is nutrition, chemical. It seems to be there are things also in the mind, in the brain that can affect this. This is something also very, very curious. And we know that, for example, post-traumatic stress disorder in parents can alter epigenetics in children. This group of authors have published this couple of papers that I believe are very, very good, and they measure the expression of that gene, and that gene is related. The monoamino acid gene is related to aggressive individuals, to ADHD, to panic disorders, to several things with behavior. And they know that when there's prenatal stress, the expression of that gene changes in the placenta, and it can be correlated to temperament in 1-year-old. And they have a way of measuring that in very young babies. And also, they've seen that it may not be only like a binary thing, wrong or bad, but also like, I don't know, a buffer situation, like it regulates a little bit. So also this group studied that in New York, there was a superstorm called Sandy. It was very impacted a lot in the population. And when mothers already had a little bit of stress and they were used to it, it buffered the effect in the expression of the genes in their pregnancy. So as I would say, moderate things are, let us say, a bit good. So not too much, not lack enough, equilibrium as always. And not everything is bad. We also know, and I will talk a little bit more of that later, that you can do intervention, and that epigenetic changes can improve, can get better. So in trauma-exposed mothers in Kosovo, there are studies that say that when you make psychotherapy and intervention in those families, it changes their epigenetic changes. But it's not only going to be, the pregnant woman, the poor pregnant woman always, being fault for everything, and the male parent is eating and he doesn't care because that has nothing to do with it. Not at all. There are some also preconceptional things. So this is even more radical. So it's not only being pregnant, but prior to pregnancy, we know that also this can affect the results in your offspring. So we have information about the methylation of DNA in the sperm of obesity in obese male partners. We know that interferes with the embryo growth during treatments, IVF treatments. You also have to know that there's a lot of information about how epigenetics may affect IRT and IVF. And we'll talk about egg donation at the end of the presentation, I promise. And then we also know about opposite epigenetic signatures in human sperm depends on fast food and healthy food, a bit like what I was just saying, and also in behavior, right? So parents that are more stressed, father and mother, also change the epigenetics in the very small children, all right? And if we're talking about preconception and we're talking about things that maybe are not related to the exposure itself of the person itself, but the germ cell prior to them, then this starts changing how we believe about epigenetics, about evolution. I'm sorry. So, classically, we know Darwin and we know that natural selection is the completely known way of making evolution. So it's just random. So if you're lucky enough and you're tall enough, then you will reach things. And if you're not lucky enough, you're small, you won't reach them, then your species will, not survive. All right. So, and Lamarck was this other scientific, a French one that prior to Darwin said, it's not like that, it's an adaptation. But he said it first. And if a giraffe, and this is the famous Lamarck's theory with the giraffe, tries and to reach for the higher part of the trees, their offspring will eventually have longer necks. And for a long time, Lamarck even ended his life very bad. He was ridiculized. And after Darwin, nobody believed in nothing like this, and everything related to this kind of theories was absurd. But there is a lot of evidence that seems to be a little bit like that, at least in some species, in some cases, in very particular moments. So it's like adding to the whole theory of evolution, right? And there are a lot of examples in a lot of model animals, and we use animals because their lifespan is shorter. So we can see other offspring very soon with a big amount of offspring. It's so much easier to make studies. It's so much more difficult to do it in humans because it will need to take a long time, also because of the ethical issues about it, right? But I'm just going to mention just one that for me, I think it's a beautiful study done in Columbia University in the United States by Dr. Marlin. And it's a study where they grabbed some rats or mouse and they took them out of their box or of their cage and they put them in another cage. And at the beginning, they put an odor, a specific odor, almond odor. And they used odor because they know that there's a specific neuron, there's a specific sensor for each odor, all right? So they apply this odor. After that, they shock the animal. So it's a bit painful. And they did that. They stressed the animal, shocking it several times a day during some weeks. After that, there was a cleaning period. And those mouses that they were all males, all right, male mouses, they were to mate with females that didn't have had that process done, and then take him along with their cage. And then they had offspring, and they sacrificed these little mouses, and they examined those neurons, and they tried to see what happened with those neurons. And they realized that it increased quite significantly the amount of neurons for that specific odor. So somehow dad, with its sperm, told the children that they need to be careful with that odor, to be more sensitized to it, because it's a dangerous thing. And that didn't talk at all. It had anything to do with that baby. So it's not nurture, it's nature, right? So finally, how does this affect egg donation? Everybody knows about this. We have a donor. She gives her egg. We put it together with the sperm of a male partner, and the female partner is the recipient of the embryo created. All right, and we actually don't know exactly how epigenetics affect egg donation or the other way around. We do have information about IVF and in general ART, but we're not so sure or so certain about how egg donation works. But I think we can discuss a little bit about this, and we can speculate and suggest some things that could happen. So first of all, we need to think that the donor, that egg, has some epigenetics changes, right? Because underwent a mother and a grandmother, all right? As I said before, the Russian doll thing. So some epigenetic things might be in that egg that is coming along, all right? Then we talk about the parents' epigenetics, the male and the female parents, and how they can affect that new embryo. and I will talk especially about the mother, because the mother during the pregnancy will affect and will, through all the evidence that I just mentioned, for sure, somehow it will change what the genome that is provided, it will change and will make its own epigenome. And that epigenome, that means the genome with epigenetic changes, the mother just affecting that, and those change those things. Also important to know, and we might use this in a close future, is that these epigenetic things might change treatment results. So we might choose, for example, a different donor for epigenetical changes, more methylation, then we choose another one, because of the results that we may have, because of the problems with epigenetics, then we may have better results of our ART treatments. And obviously, and the most important thing is the DOHA, right? That we need to know that we will improve, that we can change the epigenetics condition of our children, of our offspring. And there is also evidence for that. It is true that there's not that much evidence, and there are a lot in animals, not that much in human, but we know that food, types of food, might improve, might make those epigenetic patterns better. And we know that that gene that I just said, the one that was dangerous and aggressive and all, can be hypomethylated when we have the psychotherapy. And in panic disorder patients, we know that moderate amount of maternal folic acid in mice also improves the methylation process, and also probiotics. And this also in animals, in C. elegans, in special kind of worm. Also, it seems to improve epigenetics patterns when the microbiome, what they are eating, I'm talking about living things, microbiome, it improves also. It's very famous right now, the microbiome gut. So for that question, that is the one that for me at the beginning made me interested in this, that I heard the first time with Dr. Fabio Cruz when I was starting this fertility process. And that question that is, do I bring something to the table? Is there something of me that with the egg donation that's part of me? And I remember Dr. Cruz saying, well, yes, well, there is a very likely a chance that you do that because of epigenetics. And I thought, this is just merchandise. You're just trying to get the patient. And now we know it's completely true, right? So yes, you do bring something to the table. And last, this phrase I think is important from Lamarck, from this scientist about started everything in these evolutionary things, because we are in a moment that a lot of information we have, it's around, and a lot of misinformation. So it's a good thing for European Fertility Society, my IVF answers to have this kind of platform, so we can propagate and we get recognized the good information, the truth information. So thank you very much.
Thank you so much. I'm here, of course, don't worry. Thank you so much. This is, as I thought, of course, and of course I've seen the presentation beforehand, and it's a very fascinating topic. Thank you so much for diving deep into this matter. It's really, really interesting. And now, of course, we will have time for your questions. We do have time for your questions, so fire away, anything that's on your mind. We will start our Q&A session. Right now, there are some interesting as well questions right here. First, question from Jennifer: is it whether you can recommend a text on epigenetics, a paper on epigenetics? And she mentioned that I am a genetics graduate with a somatic form of blood cancer and want a second child via IVF.
All right. We're talking about text. There's always, two ways of looking at this. I believe that for you, I have it. I'm sorry, I have it upstairs because I could show it to you in the camera, but I don't have it in the room currently. But it's Introductions to Epigenetics. I don't remember the authors currently, and I actually used it because, as I said, I'm not a specialist in this specific mechanism, so I had to did a little bit of investigation. But I believe there's a book that I do have here that for me was very good to read about. All right, it's from an anthropologist, a paleontologist actually, and they're researching about data that tell us how the evolutionary process, how epigenetics work in the past for evolution. So it seems, just to make a brief thing about this book, that the slow processes are mediated through Darwin mechanisms, through natural selection. But acute processes, like, for example, the famous 65 million years ago meteorite that make the dinosaur extinct. So those big things, the changes that need to adapt very fast in living beings, is mediated through epigenetics. And for me, that was quite interesting, and I read it very. I think it's a good book just to read. But I believe that someone that has a lot of knowledge, it's very superficial, right? So I actually would recommend for just general population.
Okay, hope that helps.
Right.
And Jennifer has added
That helps.
here that she would like to get some details as well. So I would be happy to forward your email address, Jennifer, to Irima and the team,
I,
and of course, Dr. Zamora. And I'm sure they will be able to provide you
I have the
some
papers. I have
details as well.
all the
Exactly.
papers, so I will send them. Very good.
Perfect. I will forward the email, so don't worry, you will get the information as well. All right, let's go to the question from Lela. So, can endocrine disruptors affect epigenetics of your baby in utero and then your grandchildren as well, as you said? Yeah.
All right. We have to understand that this matter, as I said, it's very controversial, and I don't know how to say, but we have like aspects of it, right? We don't have like the whole thing completely standardized, and we know exactly how the inheritance thing works all the way along. So it's like traces. We know a little bit at the end in this kind of situation. We know a little bit this here, a little bit there, and it's a little bit like that. But we do have a lot of information on how, for example, bisphenol and BPA, how that affects an epigenetic thing. It's a disruptive, similar to estrogens, and it does change negatively the epigenetic factors and increases methylations. And so yes, there's information of that. Probably not enough to tell you that it will affect for sure your baby and your grandchildren. All right. So, but we can assume that that possibility is there. All right. So yes. I also want to say that, and I take this. Let's understand that this is a very complex process and that can be changed in the lifespan. So not necessarily just because a single particular process in a very concrete moment, it will for sure make you diabetic or obese or hypertensive just because. It'll give you an increased probability, but then you have to eat bad, then you have to, not making exercises. And it's, for example, that famous phrase that I don't know what happens to me, that I try to exercise a lot, but I don't know how to lose weight. Well, maybe you have that predisposition, right? But it doesn't mean that you necessarily are going to have issues because you are exposed a little bit at this thing at a certain point.
Okay.
All right. I hope that helps. Right?
Of course, if you have anything else, you know what to do. And also, I see another question here from Nicole, who also asked for an email with some details and the links. So I will again forward this information, and we can, and I'm sure Dr. Zamora and his team will be able to help you a bit more with this. But let me go to Larissa's question now. How does the body with a chronic inflammation like endometriosis and adenomyosis affect the effect of epigenetics? And we are thinking about egg donation after 10 transfers without implantation. Anything you can advise here?
All right. I will try to read it again. It's a long one.
Yes, sure. I can show it to
Yeah.
you as well.
Yeah, I have it there. Thank you. Thank you very much. Yeah, great. All right. For the first part of the question, it's a very good question. There are some papers, not a lot, but there are some papers, especially related to endometriosis and how there's an epigenetic pattern in patients with endometriosis that control patients do not have. So it's also another theory, and also it could be another answer to why some people have endometriosis, why others don't, and we don't have a very strong answer for that, but it seems to be also related to epigenetics. I also tell you that there are even more papers about PCO, all right? So PCO is also very related to epigenetic things. And the PCO is related to epigenetic changes in the offspring. So that is also information that we have. Um, I have to say for the second phrase more than a question that I am a I am a very, I don't know how to say this. I am egg donation for me. It's something very important, and it's very important because, for example, adoption is not that easy to get, and sometimes it's even more difficult. And what is a fertility doctor speaking about adoption? Because what I believe is that there are a lot of things that can improve with egg donation, and there are a lot of good aspects of it. And the moral aspect of egg donation is, for me, it's a bit sad when it's like it's not going to be mine. That feeling that, will it look like me? I want it to look like me, and I think it's like, I don't want it to look like me. I want it to be healthy. That's why epigenetics is important, because you can make it healthy. For me, it's very shallow to want it to be my genes, to look like me. And I understand that. I understand that you may have but maybe we need to switch a little bit that aspect. So yeah, egg donation, it's a marvelous thing to do. Actually, there's a paper in ART that describes the amount of more demethylation, DNA methylation, or more epigenetic worse patterns when we have IVF embryos out of the couple, in comparison to embryos created and with biopsy of trophoectoderm embryo, with biopsy made of donated embryos from sperm donors, egg donor. So we compare both embryos, and the embryos from the couples have way more epigenetic changes. And as I said, epigenetic changes usually mean worse things, all right, in adverse problems. So it's another good thing to say, listen, maybe I get an egg donation, and those epigenetics changes that I have had during my lifetime, I will improve through changing one germ. So
Okay.
yeah, I would obviously invite you, Larissa, to try with egg donation. It has such good results, so it's a very good thing to do, of course.
Thank you so much. Indeed, once more, very interesting here. Lots of questions, okay? And again, someone asked for some details. Cassiani, I have just made a screenshot just to have this, but of course, I would be happy to forward your email for sure. All right, let me go to the next question. There are Laura who would like to know, and I will show it to you. You mentioned there is limited evidence on the role of epigenetics using egg donor as a recipient of an egg donor. This is a little bit disappointing. How might we study the role of recipient on the epigenetics of the future child?
Yeah. I don't know how to say this in English, but Laura, she got me. She realized that I talked a lot about epigenetics, but actually there's little evidence about egg donation and epigenetics. Unfortunately, it isn't. There could be ideas of making studies to trying to find this out. We could try to match, for example, preservation of fertility. So patients that have themselves had a preservation of fertility done, and trying to compare that from an epigenetic point of view with donors the same age, the same aspect, same phenotype, and trying to match somehow that way. But it's actually it's a difficult thing to study. And because to design a study like that, I don't know. I will actually, as I said, I'm a gynecologist. I will try to talk with an embryologist, a biologist, to say how can we try to design a study that we could actually have a conclusion if the egg donation, the recipient, for example, questions like how much does it change? Does it change more when I have this external egg and that epigenetic change that I can improve? Is a very small amount of it, or it's something extremely mild and something very minimal? So those things we still do not know. But as I said, it's an ongoing thing. So there are a lot of studies. Problems. It's easier to make those studies on animals, on model animals. and they're actually making studies with IVF in model animals, but especially they're doing it, the scientists that are doing so are doing to try to take away the nurture thing, like if they mate and they inbreed and they're all the time with the animal, so something of behavior and things can affect that. So it's like even more strict and more you take that away nurture if we do just IVF, and no mating, even no mating, Right.
Okay.
Sorry about that,
Actually, I
Laura.
will let me show you the next question here. Okay. So how do epigenetics compare to nature versus nurture? We know the environment a child grows is massively affects their character, the kind of person they become is epigenetics, not just nurture.
Yes, very, very good question. And this is always something that, as a matter of fact, the ones the side of science that is still a bit contrary to these theories, it's exactly what they're saying. It's like there's nothing very weird about this. It's just nurture. It's just environmental things. And what I have to say from the epigenetic position defending it is that there is quite a lot of evidence, especially I repeat, in model animals, that it has nothing to do with nurture and the animal itself. There's plenty of information regarding C. elegans, that it's like a very special worm because we know exactly how many cells it has, how many neurons does they have, and they have done a lot of things in those worms that get inherited for quite a few generations, and without and taking the absence of nurture in that area. So I would have to say it's very, very well more established in animals, but from just a logical point of view also, we're not talking about metabolism. We're not talking about endocrine things. We're talking about genes that get expressed or do not get expressed according to external effects, external environment. So yeah, it's a little bit of those, like always, but there's a nature thing, a very genetic thing.
Mhm. All right. Once again, brilliant. Thank you so much indeed. Helen, I've seen your questions. There are quite a few of those. So I think that better is just simply to forward again to the team so they can provide you with further details. And now I will show you the next question from Claire. Okay, so I heard before there had been studies in prison about violence persons and the chromosomes they have. So if it's a bad mix, could we control how the baby is, or if it's a good family, would the kid be more likely to be?
I get it. All right. I'm not really sure about this study in particular, but the final of the question, I think we can address it. Yes, one of the things about donors, and when we are asked about donors, is we have a good criteria, and we try to be very strict to finding donors that have a certain I don't know. I don't want to be politically incorrect. I'm sorry for the language, but like a certain standard, and, and it's not only phenotype, or it's mainly phenotype things. When I say phenotype, I'm thinking about morphic external changes, but also things about behavior. Donors undergo psychology consults, and they can be ruled out if we believe there is something. But it is true that there's a part of the thing that is an interview, right? That we cannot, legally go to their records if they have a history of legal things, or their mother during pregnancy had a problem, was in prison or not. We cannot access to that, right? So what I really would like to emphasize is that we can do a lot or a little, but we can do something about it for sure. There are things that can improve. So sometimes what I think and I believe and I see in patients every day is that it's like they're requesting a lot of things from the clinics, from the donors. We have to understand that the donor is someone that we can also we cannot oblige them to be a very certain way. It's somebody that is donating a cell of our body, and we have to acknowledge and accept that. And what can we do? What is in ourselves? So I see a lot of IVF patients with body mass of 38, and they're asking if the donor this and that. So do try change your lifestyles and behavior, and for sure that will be seen in your children. And even that, I also am here to defend epigenetics in the womb, but I believe that the childhood and how we're raising our children is way more important than DNA methylation and histone modification. So it's like, for example, sometimes patients get very angry when we make a C-section and it's not a vaginal delivery. And that changes. Oh, why did it have to be that way? Well, things happen, but just look forward, and that doesn't have to be the most symbolic and important thing in your life. So yes, at some point, as I said, there could be the probability that we can meet a criteria to rule out some donors because of epigenetic things. We're not there yet, but maybe will come in the future. All right.
Of course. Thank you so much. Thank you for your explanation. Makes perfect sense indeed as well. All right, let me go for the question from Lisa. Okay, so if I understood correctly, certain epigenetics also give risk of autism. How exactly does this work? Anything you can add on that?
Well, the thing about this is like when you summarize something very. There's one paper, right? There are no. I didn't find much more than just that paper. There are paper about several disorders, all right, and there's one in particular about autism that correlates that there could be more epigenetical changes in those children, all right. Not the whole thing, as I said, all right. Not we followed mom, we know what she did or what she didn't, and how that changed in methylation process, and then we see the children, we see the process of the epigenetical changes, and then that children develops a spectrum autism disorders. So it's not exactly like that, but there is some evidence, there are some studies that are correlating that there can be some changes, and those changes, it is true that sometimes have been related. Some, for example, valproic acid. So it seems to medication like that used for anticonvulsive and agents could be related, right? Could be related. But as I said, it's a thing that, for example, we also know that age in male, all right, with the as age increases in the male partners, about 45 years old, usually it also seems to increase autism. But I don't know how to say this. As I said before, it's not something that we know exactly who is going to have it, which amount of valproic acid, or exactly which age. It's So yeah, there is some evidence that some medication, as I said, valproic acid, it could be correlated a little bit to autism.
Actually, let me just display the next question here. So what about donor's age? I was told male donor could be up to 50. Is this not important in egg donation?
This is so important that I've never heard I've never heard this, right? So I don't want to say like this is impossible, right? But I believe it's very likely that this is impossible. So we usually, most donors are under 30 years old, most of them, and there are some donors that are between 30 and 35, right? But about 35, not in our facility, and I believe not in the ones that I have worked before and that I have knowledge of, use donors about 35 years old. So, and one of the reasons, just because of the results, do not think it's about epigenetics, because we're not going to have good results with egg donation if we choose a donor that is about 35. But as I said before, we know there's also, I didn't address this, but there's also called clockwork epigenetics. All right. So we know that we can say how old is a cell through the epigenetic changes. And we know that as long as we get old, we increase those epigenetic changes. It's very logic. And also, as older the other donor is, more epigenetic changes will have, of course.
Okay. Thank you so much. Checking right now, there is one more question that I think we can still answer. And of course, as I mentioned, all those requests you've put in the chat, I will be happy to forward those to the team. Okay, so not to worry. And the question is regarding net donation at your clinic. It's a question straightforward. Do you have Japanese egg donors? It's Japanese egg donors, which is definitely
Listen,
difficult to find, right?
listen. I believe that we have some Asian, all right, donor,
Mm,
and I'm not sure if I'm using the S word, all right. You know, like the donor is the S letter. I'm sorry, is what I'm trying to say. So I believe that we have Asian donor. I will be happy to talk about the team about that. But Japanese from nationality, I don't think so. I actually don't think
mhm.
so. Right? It's just,
Yeah, it's
um,
really
it,
hard.
it's it, it actually, and the other thing about this is that we shouldn't work this way. You know what I mean? It's like nationality. It is not a thing that we use to choose a donor or not choosing a donor, right? So it's a phenotypical thing, right? So here in Spain, I don't know if how it's in other countries, but you cannot choose the donor. We choose it for you, and it's like that, and it's legally like that, and it's supposed to be like that. We're the ones that assess which is the best donor for your profile. And I will tell you right up front, we don't take in consideration nationality. To no, she's from Spain and the other one is from Italy. We want no, it's not like that, right? So it's a phenotypical thing, right? And a lot of things, height, the blood type. I won't get into detail, but not nationality.
Mhm. And while we actually on this, one more question is right here. I definitely want you to see what you just said. So let me just display it. So is it strange that I want the donor from to be from the same country as me? Would it make a difference to the baby if they feel they are from the same country? This is what you mentioned, but anything you can add?
I think I just addressed that, and I will say I don't know. This is just getting into maybe philosophical things, not so medical. But I believe that we are so mixed. We have done genetic studies on the Spanish people is from where, and I'm from Venezuela, so we are so mixed that being from the same country, there might be an increased probability that having a phenotype or looking alike with sometimes you have to understand that genes might match. There are a lot of things that will match between you and the donor because we look at that is the much more like you, but probably not the nationality has to do anything about it. So you see, I'm from Venezuela, and a lot of people in Venezuela do not have this skin color and this red hair color. And if I wanted to choose somebody, if I choose somebody from Venezuela, it would probably, statistically speaking, would be completely different from what I look like from a statistic point of view. So
Thank you for addressing this once again. I think it's important to also discuss it. So, as I mentioned, looks like that was our final question, but it's been a great and fascinating session. So, thank you so much for your questions because we are here to simply help you out with those. Dr. Zamora, it was lovely to have you. You've
thank you
explained
very much.
everything beautifully for me from my perspective. So, thank you so much for your time, and
Thank you for having me here.
it's great to discuss this topic. I'm glad that we had a chance to discuss this. And of course, remember, we will be back next week on Tuesday. There is another event, so you can join us. We will talk about unexplained fertility. So if you have some more questions, you know what to do. Just sign up, and we'll be live at 7 p.m. London time. Dr. Zamora, Omar Zamora has been our guest today from Irima Clinic in Spain. Really happy to have you. Enjoy your evening and looking
Thank
forward
you
to
very
some
much.
more events shortly with you as well. Thank you
You
so
too,
much,
Caroline.
everyone. Thank you. Thank you so much. Oh, as you can
Goodbye.
see, all the comments are definitely, um, I guess that all the patients are definitely satisfied with this session. So thanks a lot and speak to you soon, I hope, as well. Take care. Bye.
Thank you very much. Goodbye, Caroline. Thank you for everything.